All right, this is part 3 of our chapter 14 on antimicrobial drugs. So we're going to talk about the antimicrobial resistance to the chemotherapy we talked about. This is just a funny cartoon. Uh, this is obviously bacterium giving another bacterium a little piece of DNA here. And it's going to be basically confer resistance to penicillin. So the ways that bacteria can evolve to basically remove or destroy the antibiotic is here. One is called an efflux pump, which is literally a pump, right, that sits on the membrane of bacteria and it can remove. So once these antibiotics get into the cell, right, they get into the cell through different channels. But then the bacteria can just pump it right back out. Okay, so it works against these. Do not memorize, the kinds of antibiotics here, but just know that we can form a pump. We can block the entrance to them, so we can modify the channel that they used to get in so that they can't fit anymore. We can, enzymes can form, so the bacteria can pick up information to make enzymes that break down the antibiotic, right? Inactivation by enzymes. Beta lactams, we talked about this in the first part, I think, or second part. Beta lactams like penicillin that have that beta lactam ring in the middle. Remember, there's an enzyme called beta lactamase that can break down beta lactams. And then we have target modification. What target modification refers to is that so this is a good example, the ribosome. The ribosomes can mutate slightly, right? The protein, so ribosomes are made of proteins. Proteins are made of amino acids, which are coded for by bacteria. So, just by chance, remember, mutations are not directed. When mutations happen, they're completely random. And so by random chance, it might be that the ribosome has changed an amino acid, a couple amino acids, and created a version that the drugs don't fit very well anymore. Okay, so target modification is literally just the mutations that happen to be advantageous. All right, so mutation. So keep in mind, there's no directed mutation. Mutations are all random, but sometimes they actually end up being beneficial to the bacteria. And since bacteria reproduce so much, the mutations, they're not rare, right? They do mutate and they can get around it. All right, that is it. So all we need to remember is how these guys can get away with, so this is the forms of resistance, right? How can this bacteria survive in the presence of an antibiotic? It can pump it out, it can destroy it, it can, through mutation, the target is different now and we can block it from coming in. What encourages drug resistance? So what encourages bacteria to gain those things, all right? And we can see the 3 here, but because antibiotics have been used for so long and they've also been misused for so long, remember the penicillin, and for a long time after penicillin was misused, like when they came out with more antibiotics, they were also misused. So especially in places that are less developed in the world where antibiotics are sort of over the counter, I can think about Mexico. I think people used to go there and get antibiotics. When you have the drug in the environment for long periods of time, so back to this sort of picture here, right? There will be a mutant. There will be some bacteria that are just going to survive the dose of antibiotic you're taking. And when those mutants survive and you kill off all the rest of the bacteria, so the different colors here are going to represent different bacteria that have different strengths or different survivability to that antibiotic. After a treatment, you're going to be left over with those bacteria that are going to be resistant. And this says a mild dose. So when you take a mild dose, when you take doses for inappropriate uses, when you take a dose shorter than you should be, those are all ways that you can create this scenario where you're going to kill off, the easy ones to pick off, the really susceptible ones, the ones that have a little bit of an edge, an advantage, whether it's a mutation or whatever it is, they're going to survive and they're going to reproduce. So what do you have, after that course of antibiotics that you did not finish? You're going to have some resistant mutants. And if these overgrow again and you take that same antibiotic, it's not going to work because these guys are natural mutants. Okay. So again, if you get antibiotics for inappropriate uses, you're just creating an environment that's going to help those resistant strains persist and grow. When your antibiotic is correctly used, you might stop it, right? You should never do this. So sometimes people might stop the regimen. They're like, oh, I have a 7-day course of antibiotics. I feel great after 5 days. Why don't I just save the last few for the next time I get sick? So that is a perfect storm for this scenario. You want to take those antibiotics as directed for as long as the course is, because that long course is to ensure that those resistant mutants don't survive. And there are going to be outdated and adulterated antibiotics out there. There's going to be counterfeit antibiotics out there. When you buy things over the internet, who knows what you're getting, right? So we don't want to use those. We don't want to use expired antibiotics because they're going to be a milder dose. The active ingredient might have been degraded over time. So these are ways that we encourage resistance, some behaviors, right? So let's talk about drug resistance and cross resistance. There are bacteria out there that are multi-drug resistant, and multi-drug resistant microbes are called, shorthand MDRs. They're known colloquially as superbugs. And what they do is basically they carry one or more resistance mechanisms, right? So let's go back to that picture. These are the resistance mechanisms. And one bacteria can have maybe a pump and a beta-lactam block, right? So maybe the pump pumps out tetracycline and fluoroquinolone, and then this blocks out beta-lactams and tetracycline and fluoroquinolones. So you have a double whammy. Now this bacteria is going to be resistant to all of these medicines and all of these medicines, right? So that is multi-drug resistance. They can build up multi-drug resistance. We'll talk about that in a second. What is cross resistance? Cross resistance means that there's one single resistance. For example, the efflux pump is a good one to think about. So you're pumping those those chemicals out, but you're not just choosing one particular antibiotic to pump out. You can actually pump out many different kinds of antibiotics, and that's cross resistance. So let's go back to that picture again, right? This efflux pump, you can be resistant to fluoroquinolones, but you're also resistant to, this guy is also resistant to tetracyclines, beta-lactams, and macrolides because of the cross resistance, right? The efflux pump pumps out all of these antimicrobials. All right. So that's what cross resistance means. All right. And then again, same picture. I keep pounding this point, but we have a collection of a community of germs, right? They use the word germs. Some of them happen to be mutated, again, just by chance of having some kind of trait that makes them resistant. Now, I talk about mutations, but there are other things that can happen to this guy that make it drug resistance, which is obtaining something called an R plasmid. We talked a little bit about R plasmids in the genetics chapter. We talked mostly about the F plasmid for fertility. The R plasmid, or also called R factor, is another plasmid. So it's a little piece of circular RNA, it's a circular piece of DNA that is picked up by some bacteria. So remember that process called transformation. Transformation is where a bacterium can actually just take up DNA that's in the environment. So let's say one bacteria just died right next to it, right? The bacteria is dead, and it was the one that leaked out this plasmid. Now, this guy that's alive is going to uptake the plasmid. Now he has the plasmid, and he has that same antibiotic resistant that the dead one has. So transformation is a big way in which bacteria can actually just get antibiotic resistance. What is on the R plasmid? So The R plasmid, remember, this is DNA, so it's going to have the information that codes for, back to that picture, the DNA is going to code for the pump. It's going to code for those enzymes. It's going to code for, that's it. It can code for these guys because these guys are sort of like mutations. But it can code for those things. So picking up an R plasmid is very beneficial to bacteria because it gives them resistance. R stands for resistance. When you take antibiotics, right, the antibiotic kills bacteria causing illness as well as the good bacteria protecting the body from infection. Now we have the drug-resistant strain able to overgrow because the competition's gone, and now we have a drug-resistant bacteria. And then here is where this transformation can happen. Some bacteria can actually give their drug resistance to other bacteria. There's another way. Okay, so transformation is one way that we can have this horizontal transfer of genetic material. The other way, remember, was called conjugation. So I'm going to put 2. And this was again mentioned in the genome chapter or the genetics chapter. Conjugation. And conjugation, remember, was with that F plasmid. Conjugation. Okay, I totally misspelled that. But conjugation is the process of where one bacteria can actually form a sex pilus. Remember that? And it's basically a bridge that attaches to the other bacteria, and the plasmid can actually be transferred, right? This plasmid here can be transferred through that pilus into the receiving cell. So that's another way. What's the third way? How is bacteria going to pick up new genetic information? The third way, remember, it was through the virus. We have an infection via a virus. This is called transduction or specialized transduction, where the virus, when it feeds in that DNA into a bacterial cell, it can also feed in DNA that it picked up from the bacteria prior to infection. The virus picks up little pieces of the bacteria that it infected before. It can then give those pieces of bacteria to pieces of DNA to a new bacterium. I'm sorry if that was really messy, but go back to the genetics chapter to look at those 3 ways that bacteria can actually have genetic recombination or pick up new information in the form of DNA. Okay, so on top of picking up a plasmid, right, which has like one resistance gene on it, there can also be multi-drug resistant plasmids, meaning on one plasmid, there is maybe information to make a pump. Maybe there's information to break down penicillin, beta-lactamase. Maybe there's another enzyme here that breaks down another antibiotic, right? So all this bacteria does is it needs to pick up one plasmid. Lucky guy, right? It has 3 resistance, basically actions, right? 3 modes of resistance on that one plasmid. So when you have more than one resistance factor on a plasmid, it's called a multi-drug resistant plasmid. And, right, scary, that this multi-drug resistant plasmid can then be shared with other bacteria through the ways that we just talked about, through conjugation, transformation, and through transduction. So in this picture here, we have the bacteria chromosome, which is separate from the plasmids. Plasmids are usually in the cytoplasm. Although sometimes plasmids can get incorporated into the genome, all right, that does happen, but normally we see plasmids out here. And we can see that when this bacteria divides, those plasmids divide with it, right? So when this bacteria undergoes binary fission, the chromosome doubles and is in each new cell, and the plasmids double and they go to each new cell. So plasmids are passed on from one cell to the next generation to the next generation. This line is referring to that horizontal transfer, that conjugation, the transduction, the transformation, right? So we can actually give a plasmid to another cell, and now when that cell has it, all of its offspring will have it. Okay, so that is the world we're living in at the moment. We have lots of resistance genes out there that bacteria can acquire and pass on to other bacteria and to their offspring, and it's a huge problem, right? It's a huge problem, these antibiotic resistant strains. Okay, the top strains that are causing a lot of issues in the medical field right now are escape pathogens. So this is the first letter in each of their names. Enterococcus faecium is the E. We have another E, Enterobacter. So they're both basically enteric bacteria. They're in the gut, large intestine. The S is for Staphylococcus aureus. The K is for Klebsiella pneumonia. The A is for, I've never said this one out loud, Acinetobacter. I don't know. It's also an issue. And then our Pseudomonas aeruginosa. So fun fact, we just used Pseudomonas aeruginosa in class, right? Today, it was an aerobe. So these guys are multi-drug resistant. They're going to escape many conventional forms of antimicrobial therapy, and they are really difficult to treat, and they're very prominent in our nosocomial infections. They cause a large number of nosocomial infections. What is a nosocomial infection? It is a hospital-acquired infection. There is a class, there is a category of infections you pick up at the hospital because you are there, right? If you think about, before I get to that, if you think about what brings you to the hospital, you are very sick, right? If you're just like the flu, the cold, you stay home, you avoid the hospital at all costs. So the hospital really has very weak, very sick people in it. And then you have this environment in the hospital because all these very sick, very weak people come in. They might have these multi-drug resistant strains on them. And then it can get passed around in the hospital, right? So if you look at Enterobacter, opportunistic infection in hospitalized people, hospital acquired, opportunistic. That just means you have a weak person. If you have an opportunistic infection, an opportunist is a bacterium that doesn't usually cause disease, but it will if you're immunocompromised, like you're sick with something else. Another hospital setting, opportunistic pathogen, nosocomial again, right? And then Klebsiella doesn't say that, but it causes pneumonia. So you want to know escape pathogens and that they are very prominent in causing our nosocomial infections. And since they are multi-drug resistant, then they're going to be difficult to treat. These are some notable multi-drug resistant strains, and this is the S in escape. It's the methicillin-resistant Staph aureus, also known as MRSA. If you are working in the health field now or in a hospital, like you have heard MRSA probably, and it's basically Staph aureus is on the skin of a lot of people as just the normal flora. A lot of people carry Staph aureus in the skin, in the nose, in sort of the upper respiratory parts of their body. But if you somehow get an infection with it, right, you're sick, it overgrows, whatever happens to, you have a puncture wound, it gets into the puncture wound. So if you have a skin or other wound infections, you can have a infection with MRSA. If it happens to be MRSA, then it's drug resistant, right? So then when you give someone antibiotics, you have to give them other forms of antibiotics out there, right? So it's going to be hard to treat. So it says that about 1/3 of the population will carry Staph aureus in our noses and perfectly fine without illness. And then about 6% of those strains are going to be methicillin resistant. So you could have, which is kind of a scary thought, right? You could have a MRSA bacteria in your nose, not even know it because you're fine, right? It's not going to cause you to get sick. It's part of your normal flora. However, if you did get a wound and that particular strain that you have gets in there and it overgrows, now you have an issue. The other one that's not actually part of escape, but I wanted to mention it, is tuberculosis. So multi-drug resistant Mycobacterium tuberculosis is one form. There's another more dangerous form called extensively drug resistant, XDR-TB. It's also called extremely. The X can also be extremely drug resistant. But this is the Mycobacterium tuberculosis. It causes, obviously, tuberculosis. We talked about this in class. It's acid fast. It grows in your lungs. It's just, we have two forms of it. This one is resistant again against the two very common drugs that treat it. XDR-TB is additionally resistant to other drugs. So it's going to be very difficult to treat someone who has the strain of XDR-TB, right? Which is going to be an issue, not so much in the United States, but in other countries where tuberculosis is very prevalent. All right, changing a little bit to shifting into basically our food source and how our food source and our farming practices can cause antibiotic resistance. So it's the same concept of how if we took a mild dose, a short dose, an outdated antibiotic, how we're basically giving our body this background and environment of this antibiotic, and we're going to select for those strains in our body that are going to be resistant. Antibiotics are now used in many, many factory farms, right? These are really large, huge farms that make meat, for instance. And they give the cattle, they give the animals antibiotics as just part of their food. The antibiotics are in their food. The antibiotics they use for the animals are thought to, number one, prevent illness. Is basically like preventative, because the way that factory farming is physically, the animals are all very, very close together. They're stressed. They are standing in their feces, right? So if you have like hundreds of thousands of cows in a very small confined area, there's going to be a lot of feces on the ground, right? And in that feces, so if you're giving, it makes them sick, right? It's not a very good environment for these cows to be in that position. They can get sick very easily. So they practice giving the antibiotic to the cow to keep it quote unquote healthy, so it makes it till market. Another side effect that they found that antibiotics do in the cow is that it causes them to get bigger faster. So somehow this antibiotic causes them to gain weight a lot faster, which is advantageous if you're a farmer and you're like, oh, this cow is growing so much bigger than the other cows are not getting antibiotics, right? So antibiotics help with that too. So you can see antibiotics are given to pigs and chickens and cows, and what's happening in their gut is that the antibiotic kills most bacteria. But then those resistant strains will go on, proliferate, make a whole new community, or a whole new population of these resistant bacteria. Well, how does the resistant bacteria get out, right? So they're in the guts of these animals. These animals are basically breeding antibiotic resistant bacteria. How are they spread, right? So in the slaughtering process, right, where they're slaughtered. I don't want to go into it too much, but that bacteria, the resistant bacteria, can be passed on to the meat or the vegetables. And if you think about the feces that's collecting here, right, in their poop, these are in their guts. So the resistant strains of bacteria can be, I'll just draw a little bit of poop, right? They can be in the poop. And then if it rains or however they get rid of the waste, it can get into the local water system. Farms are oftentimes together. So it can sort of leach its way into another farm that's growing vegetables. So you can see that the spread can be in animal products. The meat itself can be contaminated. It can be contaminating the water in this region, and then the water is used to water vegetables, and then the vegetables can carry that antibiotic resistant strain. It can be in the environment, because when the animals go to the bathroom and they poop, this poop will eventually dry and then get carried off into the wind, and then the wind can bring it anywhere, right? And then, so it spreads away from the farm. And then we're exposed to it because we eat the contaminated food, we eat the contaminated vegetables and fruit, and then now we have taken in this resistant strain, and we can get sick from it. And it can basically, like 1 in 5 resistant strains of infections are caused from food and food animals. Okay, so it's a really big problem that we have in factory farming and drug resistance. I have for you guys a 10-minute news clip on this. I'm not going to show it here, but please watch it in the module. It's really eye-opening. And we reached the end, so we're going to have a few questions here to wrap up. Which statement is not an aim in the use of antimicrobial chemotherapy? If you want to read through it, you can pause it after I read every question, but the answer is going to be remain in the body permanently, right? We don't want that to happen. We want these antimicrobial drugs to be broken down and excreted so we don't have them building up in our bodies to toxic levels. An antibiotic that disrupts the normal flora can cause a superinfection, right? And specifically, we talked about Clostridium difficile as our very common superinfection. Number 3, targeted antibiotics include all the following except. All right, the answer is going to be C because this is an antifungal. Ergosterol is in fungi and everything else is fine. Number 4, microbial resistance to drugs is acquired through. The answer is all of them. The R factors are blank that code for what? So R factors are plasmids. They are the whole circular piece of DNA. They're also called R plasmids that code for drug resistance. When a patient's immune system becomes reactive to a drug, this is called, so this might be when you say the word reactive and immune system, right? What does that point more towards? It's going to be allergy. Okay, so the immune system is going to mediate allergy, whereas toxicity is the damage to the tissue of the organ. So it's not something that the immune system does. It's what the drug specifically does itself. And then number 6. All right, so do antivirals kill viruses in the blood? No, they do not. Okay, they basically work inside the cell in some kind of metabolism, or they might block the virus from entering the cell, but they don't act outside the cell when the virus is in your blood. And that is it for this chapter.